Good day, everyone. My name is Angiatinazzi and working for CIDSC here in the Geneva office in Switzerland. My today's speech can be considered a continuation of my CDISC 2019 European Interchange speech I had last year in Amsterdam, where I wanted to stress the CDISC community on the importance of quality in data submission. My focus today, as per the title of the presentation, will be on the Define XML and the Reviewer Guide, two critical components of any SAND or SCTM or ADAM data submission package. This is the agenda, pretty simple, with an introduction where I will also quickly discuss the Define XML and the Reviewer Guide, although I assume most of you on the call are already familiar and probably authoring or making use of both of them. I will then go through some tips that you will see if they can be considered unveiled or not. So with the objective of being to stress again on quality in data submission, let's first clarify what I mean by quality in data submission. Well, this should be pretty straightforward and obvious, at least for me. Any piece, any file, any dataset, any report, any submitted piece of code, such as SAS or whatsoever, that is a part of a data submission package submitted to the health authorities, such as the FDA, should be of good quality. Quality must be guaranteed not only in the submitted data and of course in the analysis results produced, but also in the documentation we provide as a support of the submitted data and analysis results. Attention should be paid to completeness and clarity of such documentation, as the reviewer needs to be able to understand what you have done. This is applicable to the Define XML, the Reviewer Guide, the Untitled CRF or SCTM, but also to any other files that may be provided in addition. Some of you might think you can, for example, omit to comment a conformance issue in the Reviewer Guide, either a warning or an error, thinking that this is a minor issue. But this omission can be the object of a reviewer comment or a question that could require further investigation from your side, with the risk of having an impact on the approval process and its timeline. So proper documentation is of vital importance and it could be a successful factor for your submission. Try, for example, to imagine you had the receiver of such a package and see, for example, the way you explain your derivation Define XML is clear enough. The Reviewer Guide, together with the Define XML, became a critical piece in the submission, as it facilitated the communication with the reviewer. The Reviewer Guide, either non-clinical or clinical study data reviewer guide, respectively for SAND and SCTM, and the Analysis Data Reviewer Guide for ADAM, is the document that will drive the reviewer once he or she will receive the data submission package. It is probably the first file they will open when they will start looking at your data submission package. Some sections are key. In particular, the conformance section where sponsors have the opportunity to explain unresolved issues by providing, hopefully, a clear rationale. The Define XML has an incredible power, yet probably not fully exploited, as it could play a critical role in automating processes, given the fact it has machine-readable study metadata describing the dataset you submitted and their content, including the control terminology used, any method you have implemented to create new variables, and lastly, for ADAM, you also have the opportunity to describe key analysis output through another set of standard metadata, the so-called analysis results metadata. So, despite we have good CD standard in place and templates available, with increasing availability of best practices developed by industry experts, which, by the way, I recommend to read and apply, and even if you have a good metadata repository in place, there are still probably some aspects you could improve right now when you create the Define XML and the Reviewer Guide. This is actually what I will try to do by now discussing some tips to make your Define XML review and reviewer of better quality. Let's now have a look at some of these tips. So, because of the normalized data structure used by dataset based on some of the SCTM, SAND, and ADAM models, in general, that are in general one record per subject, per topic variable, per visit, observation, etc., etc. There are cases where the variable metadata do not provide sufficient details to support review and analysis. In these cases, another set of metadata, the so-called value level metadata, should be provided in the Define XML. Value level metadata enables the specification of the metadata of a variable under condition involving one or more other dataset variables. For example, describing the characteristics of the variable VS or S in the vital sign SCTM domain, such as the data type or whether control terminology is applied, depends on the topic variable vs test cd. As such, for example, the data type of the weight is not same as the body frame size. Also, frame size is making use of a code list that is not applicable to other vital sign parameters. But to all find the data set in SCTM or BDS in Adam require the additional set of metadata. And the question in general is, when do I need to create value level metadata in the FineXML? As a rule of thumb, I would say if I open a define XML for SCTM, I would expect value level metadata are by default available for all supplemental qualifier and the trial summary dataset. In the first case, it is very likely that an individual supplemental qualifier contain information of various and different type. And similarly for trial summary, each trial summary parameter requires different attributes, including the use of different external dictionary. So here we have an heterogeneous set of information that can be not described by simply using variable metadata. But coming to the question, do we really need a value level metadata for all finding dataset in SCTM or all BDS dataset in Adam? Well, the answer is probably no. And it actually really depends and may vary from a study to another. But probably for dataset like inclusion-exclusion criteria, we do not really need a separate set of metadata to describe each individual criteria, and making sure individual inclusion-exclusion criteria are described in the variable metadata with an appropriate code list would be probably sufficient. There are other instances, like the one here illustrated, where I do also think value level metadata are not adding any value. What's, for example, the need of having value level metadata to describe how arm was assigned, or the fact that you assign the value u to a known sex? Could it have been simpler to describe this situation with a method or a comment in the variable metadata? So regardless if you have the best MDR system or not in place, handling metadata has a cost. Curing them has a cost. So the more you have to take care of. One important component of the defined XML is the terminology used by each individual variable. For example, for the variable ARL in the ASCTM dataset, the variable classifying the adverse events drug relationship, it would be interesting for the reviewer to know by looking at the defined XML how this variable was collected, if it was, for example, classified simply as related, not related, or if a more granular terminology was used, such as not related, unlikely related, and so on and so on. However, there are other situations where this is not always clear, and therefore people have different approaches when a variable or a value level metadata require a code list to be assigned. In general, a variable should have a code list defined when this is specified in the implementation guidance. Like, for example, the variable race in DM in the demographics dataset in STM require a standard CDISC control terminology to be used, or when a variable collecting the CRF, regardless if the implementation guidance expects or not a standard CDISC control terminology to be used, as a preprinted list of possible terms. This, of course, applies to STM, but in general, I would specify code list whenever a variable has a finite set of values. As such, I would not create a code list for a free text variable. And with regards to Adam, the same rules apply here. And in my opinion, for traceability principle, variable copied from SDTM into Adam need to bring from SDTM the same set of metadata, including assigned code list. Another aspect I would like to emphasize for a proper code list implementation is the use of the code. Very often I see, for example, a code list assigned to visit num variable simply listing the expected visit number from 1 to x. I recommend in such a situation to specify the meaning of 1, 2, and etc. So assign a decode value. In the example of visit num, the decode will come from the visit variable. Similarly, for other situations where a variable is a short version of another, such as test CD in SCTM or param CD in Adam BDS, where the decode will come from the test and param variables respectively. Or similarly for supplemental qualifier using the contents of the variable qlabel to define the decode of the variable qnum. There are instances where instead creating a code list in the FineXML with all possible values or with all occurrences is not required. This is the case when an external dictionary such as MEDRA is used. In such a situation, simply referencing the external dictionary and its version would be sufficient. Code list defined in the CDISC control terminology may be a superset of terms used across different variable and dataset. As such, not all terms in the code list may be applicable to all variables where that specific CDISC control terminology is expected to be used. This is the case, for example, of the unit CDISC control terminology that can be used by the variable LB or SU in LB, or also by the variable CM.U in CM, and many other similar variables in other domains. So, for example, the term application available in the unit CDISC control terminology, while it is applicable for CM.U, very likely it is not for LB or SU. In such a situation, defining in define XML a generic Unicode list and assign the same to both CM.U and LB or SU would be not appropriate. The solution would be to create in define XML two different code lists, one for CM.U and one for LB or SU, where the applicable standard term from the unit CDISC control terminology would be reported as shown here in this example. The tip in this slide gives a meaning to the title of my presentation, where I have defined the reviewer guide, the define XML brother. As mentioned earlier, the define XML and the reviewer guide together can fully drive the reviewer in the data package they receive. Although they have different purpose, in some situations, the reviewer guide can complete information provided in the define XML, or can also compensate some sort of define XML gaps. For example, because of the limitation of XML, long tests may be not always readable when rendered into the internet browser, as shown here in this example. Although a big improvement was made with the recent style sheet released by CDISC in collaboration with you. If, for example, you see your derivation could be not properly read when opening your define XML, then it's time to describe your derivation outside define XML. One option would be to move the description of your complex and long algorithm in the reviewer guide, or in one appendix, or in an external file, and reference link to it from your define XML as shown here in this example. CDISC documentation should be started with the end in mind, meaning that you do not have to wait your dataset to be produced and available before starting creating the define XML. The define XML could be also seen as a technical specification document and not a simple final output. Very often I see define XML created in an area where the author simply made, for example, a cut and paste of code used in their Adam SAS program. In some instances, this might work. For example, here the derivation of percentage change from baseline is pretty standard and straightforward. So putting the code used may be acceptable and fully understandable. However, there are situations where your code may be not understandable. If the reviewer doesn't have the same technical skill you have, and also the reviewer may miss the reason why you have, for example, in this code, created a variable a phase differently depending on the value of two other variables. Moreover, simply cutting and pasting text from the specific analysis plan or referencing a section of the same, it is not sufficient. This is again not really an added value for the reviewer, as he or she will probably need to look into your submitted program to understand how, for example, in practice, you have derived, in this example, the daily average rescue medication consumption. It is instead recommended to provide as much as detail you can, using a mix of English and pseudocode, and eventually, as per previous tip, make use of the reviewer guide or an additional external document to link to, where you provide, for example, the steps needed to create this complex derivation. Last tip I would like to share with you today, it is about the need of making sure the produced define XML are consistently created for SDTM and Adam dataset, as very often information reported into the two define XML are not cross-checked. For example, given the fact Adam is derived from SDTM, one might also assume that not only metadata of an STTM variable remain the same once copied to Adam, but also one should make sure that when STTM is referenced by another metadata, this reference makes sense and it is correct. So there are a number of checks that should be also made on the Adam defined XML to verify the traceability back to STTM is correctly implemented. You should therefore make sure that if the origin of another variable is an STTM variable, so origin equal predecessor, the variable exists in the corresponding STTM dataset. Of course, any other metadata should be consistent with the STTM metadata. For example, the study metadata or the version of the meta used, very likely the one in Adam should be the same used in STTM. Reaching the conclusion now, where I want to come back to the initial discussion on quality by making last final recommendation. So in order to guarantee a good quality in the defined XML review guide, you should not consider them as an output, and as such, do not wait the end of your trial or analysis before creating them. The reviewer guide can be used as a programming notebook, where you document things as soon as they happen, while the defined XML can be also used to automate part of your deliverables. Some of the metadata in defined XML also drive some automation when data are received and processed by the health authorities such as the FDA. For this reason, it is important metadata are accurately stored because, for example, a wrong specification of the CDISC control terminology version that you used could lead to an unexpected outcome when data are processed by the FDA. With regards to reviewer guide, you really have in your hands an important communication channel with the reviewer. As such, it is important you communicate well any problem you had during data collection or during the analysis, so that the reviewer will not need to come back to you asking for more details. As a final recommendation, in order to have deliverable of good quality within the organization, you should establish some conventions, such as naming convention for code lists or wording for methods. Together, of course, with a proper review process that should be not limited to simply perform validation through some standard validation software. Thanks a lot.